Welcome back, fellow curious minds. Thank you for joining me once again. Settle in, get comfortable, and let's explore another fascinating story together. Picture this. It's May 14th, 1796. In a small English village, a country doctor is about to perform an experiment that will either save humanity from one of its oldest killers or murder an 8-year-old boy. Edward Jenner takes pus from an infected cowpox pustule on a milkmaid's hand. He makes 2 small cuts on young James Phipps' arm. Then he rubs the infectious material into the wounds. 6 weeks later, he does something that seems utterly insane. He deliberately exposes the child to smallpox, a disease that kills 3 out of every 10 people it touches. The boy shows no symptoms. Not one. In that moment, Edward Jenner doesn't just save James Phipps. He launches a scientific revolution that will reshape human civilization and save more lives than any single medical breakthrough in history. This is the story of vaccines. Not just what they are or how we make them, but how they actually work inside your body. It's a tale spanning 228 years, from cowpox pustules to synthetic genetic code, from folk wisdom to Nobel Prizes, and it reveals one of the most elegant partnerships in all of biology, the collaboration between human ingenuity and your immune system's extraordinary memory. To understand how vaccines work, we need to start with that terrifying gamble Jenner made in 1796. But first, let's understand what he was fighting. Smallpox was humanity's ancient enemy. For over 3,000 years, it had swept through civilizations like a biblical plague. The virus covered victims in painful pustules, scarred survivors for life, and killed without discrimination. Entire royal bloodlines fell to smallpox. It toppled empires and emptied cities. But in the English countryside, milkmaids told a different story. "I shall never have smallpox," they would say, "for I have had cowpox. I shall never have an ugly, pockmarked face." This wasn't medical theory. It was folk wisdom passed down through generations of women who worked with cattle. Edward Jenner was a country physician, but he was also a scientist. He had trained in London, studied under the great surgeon John Hunter, and even made important discoveries about cuckoo birds that earned him election to the Royal Society. When he heard the milkmaid's claims, he didn't dismiss them. He wondered if they might be true. Cowpox was a mild disease that cattle sometimes caught. Milkmaids occasionally picked it up from infected udders, developing a few pustules on their hands that healed quickly, with no lasting effects. But could this minor infection really protect against deadly smallpox? On that May morning in 1796, Jenner decided to find out. His test subject was James Phipps, the 8-year-old son of his gardener. Jenner took material from a cowpox pustule on the hand of Sarah Nelms, a local milkmaid, and inoculated the boy. Now here's what makes this experiment so terrifying. Jenner was betting everything on an unproven theory. If he was wrong, young James would face a 30% chance of death and certain disfigurement if he survived. There was no safety net, no backup plan, no medical ethics committee, just a country doctor, a small boy, and a hypothesis that could save the world or end a life. James developed a mild fever and a few pustules, then recovered completely. 6 weeks later came the moment of truth. Jenner exposed the boy to actual smallpox through the standard inoculation method of the time. Then he waited. Days passed. Nothing happened. No fever, no pustules, no symptoms at all. The cowpox had somehow made James immune to smallpox. The milkmaids had been right all along. But what exactly had happened inside James Phipps's body? In 1796, nobody had any idea. The discovery of germs was still decades away. The concept of an immune system hadn't been imagined. Jenner knew that vaccination worked, but he had no clue why. He published his findings in 1798 in a paper called An Inquiry into the Variolae Vaccinae, known as the Cowpox. He coined the term vaccination from the Latin word vacca, meaning cow. The Royal Society initially rejected his work for insufficient evidence, forcing Jenner to conduct more experiments. But gradually, as vaccination proved successful again and again, the medical world took notice. Within decades, vaccination had spread across Europe and beyond. Napoleon had his entire army vaccinated. Thomas Jefferson called Jenner a benefactor of mankind. By the early 1800s, thousands of lives were being saved. But the mystery remained. How could exposure to a cow disease protect humans from smallpox? The answer would take over a century to unravel, and it required understanding one of the most sophisticated systems in all of biology, your immune system. Think of your immune system as the most advanced security network ever created. It has patrol units that constantly scan for threats. It has intelligence agencies that identify and catalog enemies. It has special forces that launch targeted strikes. And most remarkably, it has a memory system that never forgets a face. This memory system is the key to how vaccines work. But in Jenner's time, nobody knew it existed. The breakthrough came in stages, like assembling pieces of an enormous puzzle. In the 1890s, scientists discovered antibodies, special proteins that could neutralize toxins and kill bacteria. They found that blood from recovered patients contained these protective substances. This was the first glimpse of immune memory. Then came the discovery of different types of immune cells. B cells that produce antibodies, T cells that coordinate attacks and kill infected cells, and most crucially, memory cells that patrol your body for decades, waiting for the return of old enemies. Here's how it works. When a foreign substance, what scientists call an antigen, enters your body, your immune system launches a complex response. Some immune cells capture pieces of the invader and present them to other cells, like showing a photograph to witnesses. B cells begin producing antibodies, proteins specifically shaped to bind to and neutralize that particular threat. T cells coordinate the response and hunt down any cells that might be harboring the enemy. But here's the truly remarkable part. After the battle is won, your immune system doesn't just forget and move on. Some of those B cells and T cells transform into memory cells. They're like veteran soldiers who remember every detail of the fight. They circulate in your blood and lymph system, sometimes for your entire life, carrying the molecular signature of that specific enemy. If the same threat ever appears again, these memory cells spring into action instantly. They don't need to figure out what they're fighting or how to fight it. They already know. Within hours, they're mass-producing the exact antibodies needed and launching precisely targeted attacks. The invader is destroyed before it can establish a foothold. This is why you usually only get diseases like chickenpox once. This is why vaccines work. When Jenner inoculated James Phipps with cowpox, he was essentially introducing the boy's immune system to a molecular cousin of smallpox. The cowpox virus was similar enough to smallpox that the immune system's memory cells could recognize the deadly disease when it appeared. But cowpox was mild enough that it didn't pose a serious threat during that initial exposure. It was biological cross-training, like learning to fight one opponent in order to defeat a more dangerous one. But Jenner's discovery was just the beginning. The next great leap came from Louis Pasteur, the French microbiologist who extended vaccination far beyond smallpox. Pasteur realized that Jenner's principle could work with other diseases if scientists could find a way to weaken dangerous pathogens while keeping them recognizable to the immune system. In the 1880s, he developed vaccines for rabies and anthrax using revolutionary laboratory techniques. The rabies vaccine came with its own moment of terrifying drama. In July 1885, a 9-year-old boy named Joseph Meister arrived at Pasteur's laboratory. He had been bitten 14 times by a rabid dog. Without treatment, the boy faced certain death. But Pasteur had never tested his rabies vaccine on humans. He faced an agonizing choice. Let the boy die, or try an experimental treatment that might kill him faster. Pasteur chose to act. Over 14 days, he gave young Joseph a series of increasingly strong doses of weakened rabies virus. It was essentially the first human vaccine trial, conducted under the ultimate pressure. The boy survived. The vaccine worked. And Pasteur proved that vaccination could work even after exposure to disease, opening up entirely new therapeutic possibilities. Through the early 1900s, vaccine science advanced rapidly. Scientists learned to create different types of vaccines. Live attenuated vaccines used weakened versions of viruses or bacteria. Killed vaccines used pathogens that had been inactivated, but still retained their molecular signatures. Toxoid vaccines targeted the poisonous substances that some bacteria produce. Each approach trained the immune system in a slightly different way, but the principle remained the same. Show the immune system what an enemy looks like in a controlled, safe environment, so it can respond instantly if the real threat ever appears. By the 1950s, vaccines had conquered some of humanity's worst enemies. Diphtheria, tetanus, whooping cough, and polio were all yielding to vaccination campaigns. The success of Jonas Salk's polio vaccine, tested on over 1,800,000 children, proved that vaccines could work on a massive scale. But scientists were already imagining the next revolution. What if they didn't need to use actual pathogens at all? What if they could engineer vaccines at the molecular level, creating precisely designed immune training programs? The first step came in 1986 with the hepatitis B vaccine, the first created using recombinant DNA technology. Scientists inserted the genetic code for a hepatitis B protein into yeast cells, then harvested the protein to create a vaccine. No virus required. But an even more radical idea was taking shape. What if scientists could get human cells to produce vaccine proteins directly? What if they could turn the human body into its own vaccine factory? This idea led to one of the most remarkable scientific stories of our time: the development of messenger RNA vaccines. In 1989, scientists first managed to inject messenger RNA into cells and get them to produce proteins. Messenger RNA, or mRNA, is essentially a molecular instruction manual. It carries genetic information from DNA to the cellular machinery that builds proteins. The concept was elegant. If scientists could create synthetic mRNA with instructions for making a pathogen's signature proteins, they could inject that mRNA into human cells. The cells would produce the proteins, the immune system would recognize them as foreign, and memory cells would form, all without ever exposing the person to actual pathogens. But there was a massive problem. When scientists tried injecting synthetic mRNA into animals, the immune system attacked the mRNA itself before it could do its job. The body's natural defenses saw synthetic RNA as a threat and destroyed it immediately. For decades, mRNA vaccines seemed impossible. Many scientists abandoned the field. Research funding dried up. The idea looked like a dead end, but one scientist refused to give up. Katalin Karikó, a Hungarian-American biochemist, believed mRNA vaccines could work if she could solve the immune recognition problem. In 1995, the University of Pennsylvania demoted her, telling her that mRNA research was going nowhere. Karikó persisted. Working with physician-scientist Drew Weissman, she spent years testing different modifications to synthetic mRNA. They needed to find a way to make the mRNA invisible to the immune system's sensors, while preserving its ability to instruct cells to make proteins. In 2005, they found the answer. By replacing one of mRNA's chemical building blocks, called uridine, with a modified version called pseudo-uridine, they could slip synthetic mRNA past the immune system's detection systems. It was a breakthrough that would change the world, but it took 15 more years to prove it. When the COVID-19 pandemic began, Chinese scientists published the genetic sequence of the SARS-CoV-2 virus on January 11, 2020. Within 48 hours, scientists at Moderna had designed an mRNA vaccine. They created synthetic genetic instructions for human cells to produce the virus's distinctive spike protein. The speed was unprecedented. Traditional vaccine development takes years or decades. The mRNA COVID vaccines went from genetic sequence to human trials in just 65 days. They were authorized for use 11 months after the virus was sequenced. Here's how they work. The vaccine contains synthetic mRNA wrapped in lipid nanoparticles, essentially microscopic fat bubbles that protect the mRNA and help it enter cells. Once inside muscle cells, the mRNA instructs them to produce the COVID virus's spike protein. The immune system immediately recognizes these spike proteins as foreign. B cells begin producing antibodies. T cells coordinate responses. And crucially, memory cells form, carrying the molecular signature of the spike protein. If the actual COVID virus later enters the body, those memory cells recognize the spike protein instantly. They launch immediate, targeted responses that can neutralize the virus before it establishes serious infection. It's the same principle Jenner discovered in 1796, but executed with molecular precision. Instead of using cowpox to train immunity against smallpox, scientists used genetic instructions to train immunity against COVID. The mRNA vaccines saved millions of lives during the pandemic. In 2023, Katalin Karikó and Drew Weissman received the Nobel Prize in Physiology or Medicine for the discoveries that made mRNA vaccines possible. But this is just the beginning. mRNA technology opens up possibilities that would have seemed like science fiction just decades ago. Universal flu vaccines that provide protection against multiple strains. Cancer vaccines that train the immune system to attack tumor cells. Vaccines for diseases that have never been preventable. Scientists are even exploring therapeutic vaccines, treatments that boost immune responses against existing infections or diseases. The same immune memory system that Jenner first harnessed could become a platform for treating conditions from Alzheimer's disease to autoimmune disorders. What makes this story so remarkable is how it reveals the power of cumulative human knowledge. Jenner's Cowpox experiment led to Pasteur's laboratory techniques, which led to modern immunology, which led to recombinant DNA technology, which led to mRNA vaccines. Each generation built on the discoveries of those who came before. Folk wisdom became empirical observation, which became scientific theory, which became molecular engineering. The Milkmaids saying about cowpox protecting against smallpox became the foundation for some of the most sophisticated medicine in human history. But perhaps most remarkably, all of this advanced technology still depends on the same biological system that protected our ancestors. Your immune system's memory cells work exactly the same way they did in James Phipps's body over 200 years ago. The hardware of human immunity hasn't changed. Only our software for training it has evolved. Think about that the next time you get a vaccination. That tiny injection is connecting you to an unbroken chain of scientific discovery, stretching back to a country doctor and a milkmaid in 18th century England. It's harnessing an immune system that took millions of years to evolve, using knowledge that took centuries to accumulate. And in your arm, memory cells are forming right now, molecular sentinels that will patrol your bloodstream for decades, carrying the signature of threats you may never encounter, standing guard with the same biological precision that has protected our species since long before we understood how it worked. Edward Jenner saved James Phipps with cowpox and intuition. Today's scientists save millions with genetic code and engineered precision. But the fundamental partnership remains the same. Human ingenuity working with biological memory to give us the most powerful tool we have against infectious disease. In a world where scientific progress sometimes feels overwhelming or abstract, vaccines remind us that the most profound advances often work by understanding and collaborating with the natural systems that already exist within us. We didn't invent immunity. We just learned how to teach it. And that's where we'll leave things for today. Thank you for listening, as always. If this journey sparked your curiosity, there's plenty more to explore, so stay tuned. Until next time, keep questioning, keep wondering, and never stop looking back to understand the road ahead.